So next, I'd like to introduce Beth Wood. Beth is a genetic counselor at the Center for Neurodegenerative Disease Research. She specializes in neurodegenerative conditions. We're very, very fortunate to have Beth here. There are very few folks with Beth's knowledge and with her expertise in this area. And this is true not just in the area, but not just true across the country, but it's true internationally. Beth is really an expert in the area, and we want to thank her for her contribution and for discussion of genetics in FTD. Beth.
It was. Thank you so much. It was a wonderful introduction. Set me up very well. So again, as Dr. Grossman was saying, my role within the Center for Neurodegenerative Disease Research is as a genetic counselor. And today I just want to take some time to, one, update you on the information and the genes that we know that can cause frontotemporal dementia and related conditions, and two, spend a little time talking about the practical applications and what we can actually do with this information. So to start off, I wanted to just give a little review of how genetics fits into the big picture of learning and treating FTD. If we have knowledge of a gene, if we know a gene that causes the disease, this gives us two types of information. One is it gives the family information. A lot of times people have questions. You know, my mother has FTD, my grandmother, my brother, my sister. What does that mean for me? Unless we know knowledge of the genetics of the condition, it's really hard to answer those types of questions. If a genetic risk is found in the family, that would give the family members information for predictive testing to find out if they themselves also carry the gene change. As well as in the future, hopefully there'll be the development of preventative measures to prevent people from ever having onset. The other thing a gene tells us is it gives us knowledge as to why the disease is happening. This can be a very frustrating question. Why did my loved one get this disease? If we know there's a genetic cause, that tells the doctor why the disease is happening and possibly gives some insight. Certain genes or certain gene mutations may be associated with particular clinical symptoms that would be helpful for the doctor and the family to be aware of. And of course, ultimately, if you know why a disease is happening, you can develop medications, you can develop cures and preventative treatments that we are all so avidly searching for. So now it's time for Genetics 101. Want to make sure we're all on the same playing field. If you take any cell in your body, there we go. Your genetic code is stored in every cell in your body. It's stored on these little structures called chromosomes. And basically, if we follow this diagram out, what a chromosome is it's a tightly wound up coil of DNA. So here's the double helix that we're probably all familiar from seeing from the newspapers and news programs. So if you can think of a chromosome, basically what a chromosome is a strand of thousands of genes. A gene is just one particular area of the DNA code. What a gene does, it's basically like a recipe. The body's able to read that gene, and it tells it how to make a protein. Proteins are ultimately what our body needs to function correctly and have the right structure. Now, when we talk about disease and having a gene for a disease, what that means is not that a person has a different gene than the rest of us. What it means is there's a change in that gene. Their gene has a mistake or a change, and usually you hear the word mutation. You can see here in this example, we have the normal gene. It has the right code in the genetics, and it makes the right kind of protein. One type of mutation that we see is when part of the DNA is deleted. When there's a loss of part of the recipe, part of the gene, the protein that results is either going to be incorrect, or this gene will cease to function and it won't make any protein at all. And that is what leads to genetic diseases occurring. So going back to chromosomes, we all have 23 pairs of chromosomes. Chromosomes come in pairs, sort of like shoes or gloves. You get one set of the pair from your mother and one set from the pair from your father. Males and females, for the first 22 pairs, this is just an organized layout of the chromosomes. The first 22 pairs, they're equal in males and females. We all have the same chromosomes through 1 through 22, and we all have the same genes on those chromosomes. The only thing that's different between males and females is that a male has an X and a Y, and a female would have 2 X's. In this case, I've circled the pair of chromosomes number 17 because I'm going to be talking about them later. That's actually, these chromosomes is where we know the genes are that we know of that cause frontotemporal dementia. So, getting back to FTD, if we take a look at all the families, all the patients, and we look at their family history information, we know that the vast majority, about 60%, would fall into the sporadic category. What sporadic means is that there's only ever been one person in the family diagnosed with FTD, and it's very unlikely that there'll be a genetic cause or explanation for their disease. It's most likely not hereditary. About 30% is something called familial. What familial means is that there's at least one other person in the family that's been diagnosed with either FTD or a condition related to FTD. And then the last section is about 10% of our families we would categorize as hereditary. And what that means is that we believe there is a gene mutation that's causing the disease to occur. These, in this 10%, the majority of that 10% of hereditary FTD is caused by either the tau gene, which is also referred to as MAPT, which is on chromosome 17, as well as the progranulin gene, also found in chromosome 17. I would just like to mention that it's believed that it's just a coincidence that these 2 genes happen to be on the same chromosome. There's no known genetic interaction within the chromosome. Mutations in either tau or progranulin the way they are inherited is in something called autosomal dominant inheritance. So getting back to inheritance in chromosomes, in this example, imagine this is the pair of chromosome number 17. In this example, the red box, that's marking the gene that's affected, that has a mutation. In the gray box, that gene is fine. It does not have any changes. When this individual, since they have a mutation, they will be affected with the disease. For inheritance, anytime an affected individual with a gene mutation has a child, there's a 50% risk of passing on the disease gene and an equal 50% risk of passing on the non-disease gene. This risk is equal for men and women. There's no gender difference. And if you follow it down, an individual who does not inherit the gene change, they are not at risk for the hereditary form of the disease, and it's not possible for them to pass on the gene mutation when they themselves have children. The gene mutation is gone from this branch of the family. It can't skip generations. So to talk a little bit more about tau, I'm sure a number of you in the audience have either heard talks on tau before or read information about it. What the tau gene does is it tells the body how to make the tau protein. Just wanted to explain quickly what the tau protein does. If you think of a neuron in the brain, it sort of has this 3-dimensional shape. It has a large body and a long tail, and it needs to keep this shape in order to function correctly. If you think of the cell as sort of having a skeleton, the skeleton or the scaffolding would be made up of this component called microtubules. The tau is part of that skeleton. It helps stabilize the microtubules so the neuron keeps its shape and can continue to function. So if there's a mutation in the gene that tells the body how to make the tau protein, that's going to lead to problems in the onset of the disease. Sometimes, very often, you'll hear that if an individual has a mutation in the tau gene, this is referred to as FTD-P17, which stands for Frontotemporal Dementia with Parkinsonism linked to chromosome 17. And this brings up an important part about the clinical features of an individual with a tau mutation. Not all, but a significant portion of families that carry a tau mutation, you'll see Parkinsonian features as well as the frontotemporal dementia. There's typically a younger age of onset if an individual has a tau mutation. Typically, the average age of FTD is somewhere in the ballpark of 60, but if an individual has a tau mutation, the onset's usually between age 40 and 60. There's a wide range of the FTD cognitive and behavioral symptoms seen. There's no single clinical symptom that makes us suspect or know that an individual has a tau mutation. If an individual with a tau mutation comes and has an autopsy, what the pathologist will see when they look at the brain cells is an excess accumulation. I think Dr. Coslett was talking about this earlier. They'll see excess tau in the brain cells. This is referred to as a tau-positive autopsy finding. So next I'll talk about progranulin. Progranulin is actually fairly new. It was just discovered last summer, so we've been learning about progranulin over the past year. Again, it's also on chromosome 17, and what the gene does is it makes the protein progranulin, which is an important growth factor that's naturally found in many different tissues in our body. At this time, we do not yet fully understand what the connection is between the loss of the normal progranulin function and the onset of FTD. That's something, obviously, that researchers are actively working on. One of the most interesting features of progranulin mutations is that there's actually a very wide range of onset. Depending on the studies, as well as our studies here at Penn, the onset age can range anywhere from the mid-40s to the early 80s. There's, again, a wide range of clinical symptoms. There's no single clinical symptom that makes us suspect a progranulin mutation. One thing that has been noted is that it's very rare to see symptoms of motor neuron disease in individuals with progranulin mutations. In this case, when we look at autopsy findings, they do not have excess tau in their brain cells. Instead, it's something called tau negative, or one of the specific types that Dr. Grossman talked about is FTLDU. This is actually a very exciting finding that a lot of work is being done on, because here at the University of Pennsylvania and Dr. Lee and Dr. Trzynowski's lab, we now know that the disease protein that's in FTLDU is TDP43. And again, you're probably going to hear a lot more information about that in just a bit. There are some other rare genes that have been associated with FTD. I'm not going to take a long time going over these. They've just been found in a handful of cases across the world. One of interest that I do want to mention is this last one, which is presenilin 1. This is actually a gene that causes early onset hereditary Alzheimer's disease. The reason I bring it up is that occasionally, it can be hard to know if someone has frontotemporal dementia or maybe a frontal variant form of Alzheimer's disease. So in these cases, if we have a patient that has a strong family history of FTD or a strong family history of FTD-like symptoms or Alzheimer's-like symptoms, and we don't find a change in progranulin and we don't find one in tau, it might be worth taking a look at the presenilin gene to see if there's a change in that gene. In about 5 to 10% of ALS patients, they also have a diagnosis of FTD. At this time, we do not know a gene that causes both ALS and FTD. It's a very active area of research, and the researchers have actually narrowed it down to a particular region on chromosome number 9 that they think the gene is located in. You can think of it sort of, they know the city the gene lives in, but they don't know its street address yet. I can assure you this is a very active area of research, and I'm hoping that they'll have some information for us, an actual result in the very near future. I briefly just want to touch on ALS. Now, this is straightforward ALS, not at all connected with FTD. We know that approximately 10% of ALS patients have a positive family history, at least one other person that's been affected. And there is a gene that we know of called SOD1, and we found that about 2 to 5% of ALS patients will have a mutation in SOD1 that was the cause of their disease. So it is rare, but there is a clinical genetic test available that if you know someone or you're in a family that you want more information about SOD1, testing is available, and you can talk to either someone such as myself or your doctor about it. So coming back to my pie graph, as you recall, I said we have this 30% that's familial. They don't fall in this hereditary category. So we're not sure yet if there's a gene that's causing the FTD, but we still see more FTD in the family than we would expect by a general chance. It can be very frustrating to be in this category because we can't give, at this time, we can't give exact risk numbers to other family members. One active area or one active theory is that this type of familial family history might be due to something called multifactorial inheritance. And what this means is basically is families have a very similar overall genetic makeup, may share several common genes or maybe minor gene mutations. And they also very frequently share the same kind of environmental factors, the places they live and have grown up, their eating habits, overall lifestyle habits. So there's a theory that if you have a combination of these, that a combination of being susceptible to FTD, as well as having certain environmental triggers, they could combine and cause the disease to occur. I'd like to stress that at this point, we don't know of any environmental triggers for frontotemporal dementia. This is something that here at Penn we're actively researching. We have an amazing packet where we try to document any single kind of environmental trigger so that we can go back and look and see if there's any that are really common between FTD patients. So first we need to figure out possible environmental triggers, and then we can start looking into this combination. So we've made it through the real scientific, the review of the genetics of FTD. So now I'd like to take some time to talk about, what does this information really mean? A lot of times I get questions, can I get a genetic test? Should my family consider genetic testing? And if we should, how do we go about doing that? What options are available? How do you go about getting a genetic test? To determine whether genetic testing is something that's either appropriate or something that's worth looking into, the most useful tool we have is the family history, which we often refer to as a pedigree. In this case, just to orient you, squares are males, circles are females, and anyone with a little blue square is someone that has a diagnosis of FTD. In this particular family history, you can see that there is a very strong history of FTD. It goes from parent to child, and then parent to child, and passed on through the bloodline. This is a great example of that autosomal dominant inheritance that I was talking about. This is an example of a family that may very well want to consider genetic testing or just get more information so that they're well informed about their options. However, the majority of cases are not that clear-cut. This is an example of a familial family. We have our patient down here who has a diagnosis of FTD. Both her siblings, both fine, they haven't developed any symptoms. Her mother lived to 91, no symptoms, and her father died at 81 with no symptoms. However, she has a paternal uncle that he reportedly had some kind of dementia. We don't know what it is, so it's marked differently. But the family remembers him having personality changes, and it seemed like he had some similar clinical features to our patient. This is an example of a family where it's not so straightforward. It's important to learn more if the family is interested in genetic testing. There's obviously options available, but again, it's possible that we won't find a gene mutation, and it's going to be very difficult to tell her children, tell her other family members what their exact risk is. I've been talking a lot about genetic testing. There's actually two types of genetic testing, clinical and research. I found that this is a very confusing area for people, so I want to take a minute to just explain what the difference is. Clinical testing is like a regular blood test that you can go to your doctor, they can order it, send it off to a professional lab, and they report back a result. There's usually a charge for the test, and very often medical insurance does not cover genetic testing. There's a guarantee of results. Usually in a matter of weeks, you'll get back a result telling you whether the result was positive, they found a gene mutation, or negative, they did not. And the written report is something that does become part of the medical record. It has the patient's name, date of birth, medical record number on it. Research testing is something very different. There's two purposes for research testing. One is to better understand the condition, and two, learning more information about the gene so that we can develop the regular clinical test that people can order. There's usually no fee. Research testing is free. However, there's no guarantee of results. There's a lot of controversy between research centers whether or not it's appropriate to give back a research result. One of the reasons is that we don't track a patient identifiers with that sample. They're given a code number. So there's no patient name, date of birth, medical record number tracking the sample throughout the testing process. Because of this reason, a research result should not be used as a clinical result. So, clinical genetic testing for FTD is available on a limited basis. For the tau gene, there's about 3 to 4 labs, University of Penn being one of them, that do provide clinical testing for the tau gene. The techniques that they use for the testing, the types of tests, they do vary, and of course the pricing varies. If this is something you're interested in more information about, I highly recommend you can contact me or someone that's more local to you. I'd be happy to help you out so you can discuss those options in more detail. Progranulin, there's not clinical testing yet in the United States. As I was saying, we've known about the gene for about a year now, so much of the testing that's going on right now is research testing. We here at the University of Penn, we realize how important it is to have a clinical test for progranulin, and it's actually something we're currently working on getting set up, and I'm hoping that we have the clinical test within the next year. So what are the reasons that someone that has a diagnosis of FTD might want to consider getting a genetic test? One is that if their test result is positive, it really strengthens the diagnosis. It gives us a really good indication that, yes, this is frontotemporal dementia. It can possibly give insight into the disease prognosis. As I mentioned before, certain mutations are linked with either certain clinical features or certain features of the disease that it might be very useful for the doctor to know about ahead of time. Depending on the genetic status, it might affect individuals' eligibility for certain clinical trials. Obviously, if you know that there's a gene mutation in the family, that's an incredibly powerful piece of information for giving risk values and genetic counseling for the at-risk members in the family. And lastly, in the future, depending on an individual's overall genetic status, their genetic findings, that actually might be very powerful information in determining their response to specific therapies and treatments. Regarding at-risk individuals, people who do not have symptoms of FTD but know they have a family member with a gene mutation, there's also genetic testing available for them. It's a different process, though. We often call this either predictive, or sometimes you'll see it called pre-symptomatic genetic testing. In this type of testing, you can't just go to your doctor, put your arm out and say, draw my blood and tell me my genetic fate. There's actually very well-documented guidelines for how to approach this type of testing. We always do genetic counseling and some form of a psychological evaluation before and after the testing process. Genetic testing of children, anyone under age of 18, is not recommended for an adult onset condition. The types of things that we talk about in genetic counseling. One, if someone's coming in and they say, okay, my mother, I know she has a gene mutation, and I think that I might want to find out my genetic status. One of the first places we'd start off with is education, making sure that all the person's questions about how genes are inherited, what their exact risk number is. Want to make sure all that information is clear. We talk about motivations for testing. Why have you decided at this point in your life that this is information that you want to pursue? The reasons can vary, and it's something just good to talk through and feel comfortable with your decision. We discuss possible outcomes, the positive result versus the negative result. Oftentimes people just fixate on one, and they're sure that they're going to be positive, that they have the gene mutation. And they never take the time to think about how they would react if the test result is negative. And then lastly, the testing process is really set up to be another form of both psychological and social support. Finding out one's genetic status obviously can be a scary thing, and it's a big decision. So this testing process isn't set up to decide who can get testing, to allow someone to get testing. It's really for the counselor and the team to be there as another support for the person as they go through the testing process. Oftentimes, the decision comes down to weighing the risks and the benefits. Some risks of knowing genetic status is one, emotional. If the test result comes back positive, there is a gene mutation, this person obviously can be at risk for depression, anxiety, and there is a concern of suicide as well if that person does not get help to deal with their depression and anxiety. However, there's also a risk to getting a negative result, not having the gene mutation. We call this actually survivor guilt. The person feels guilty. This is very common. They feel guilty that they escaped the disease that's going to affect other family members. Another big risk that I hear quite a lot about is discrimination. Insurance discrimination, employment discrimination. There aren't a lot of well-documented cases of genetic discrimination, but it's obviously a concern. And if you've been reading the papers, you'll have seen that there is a bill currently going through the government and hopefully be signed into law to provide protection for genetic privacy. The benefits that someone may mention for genetic testing: one, life planning. If someone knows that they're positive, they know that they're negative, they can make decisions such as, for instance, a lot of times if people know they're positive, they'd want to do finances and make sure all their financial and personal matters are in order. Prenatal decisions is actually a big one that I hear about. People want to know if, I want to know if I carry the gene mutation so that I know if I'm at risk to pass it on to their children, to my children. And there are actually now prenatal options that individuals can take advantage of that either lessen or eliminate the risk of passing on the gene. And then lastly, a lot of people they feel a huge benefit of needing to know. They're anxious, they're worried, and they can't live with that waking up every morning wondering what their genetic status is. So lastly, I just want to close that again reminding you that the genetic research that's being done on FTD, ALS, and this whole field of conditions is extremely important. Whenever we find a new gene mutation that's linked to disease, it allows us to give information to the family members that the family members are looking for. It gives the scientists and the doctors the information they need to develop better medications and cures. And lastly, along with those medications and cures, hopefully they'll also develop preventative measures so that at-risk individuals never have to experience onset of the condition. So lastly, I'd just like to close with putting up my contact information. This can be a sensitive topic that people have more information about. And also, we here at Penn are very lucky. We actually have 2 genetic counselors that are solely specialized in researching neurodegenerative conditions. And that's Dana Clay, who both Dana and I will be running the breakout session on genetics. So please bring your questions, and I'd be happy to answer them. Thank you.